He said he put a smoke stick up there to get the airflow from the HEPA filter down to the ground inside Colorado, where he's at in Fort Collins. And it came around the other side and it infected this negative control bat cage above. This thing was so contagious, there was no containing it. This thing would have leaked from a BSL-5. They threw the kitchen sink at this thing, not to infect us humans. It's an accident. It's an animal vaccine to infect live bats. This thing, the R0 was cranked up, and that was shown in what I consider to be the smoking gun email between these two.
Okay,
so unknowingly gave it up.
Welcome to Manifold. The interview you're about to watch is with Jim Haslam. He's recently written a book on COVID origins, and he has dug extensively into the information that emerged since the pandemic started in 2019. Now, one of the things I didn't emphasize enough in our conversation, because there's so much information to process, and Jim raises so many both interesting and speculative points in the conversation, is simply the following. The evidence now is strong that prior to 2019, the United States had the capability to genetically engineer a virus very similar to COVID-19. So whether or not the virus was engineered, the actual virus which killed millions of people, whether that really was bioengineered or leaked out of a lab, is somewhat secondary from the scientific point that we do now have the capability to construct such viruses de novo in the laboratory. I think the evidence is strong now that we can do that, and that there are researchers working on this kind of thing, funded by US taxpayer dollars. At the time when the pandemic started, it wasn't clear at all, at least to me and most of the people, all of the people that I talked to, whether this really was scientifically possible. I think a few virologists, experts at engineering coronaviruses, knew the answer then, but we as a general population didn't have access to that information. Even other scientists didn't have access to that information, that this was indeed technically possible. So aside from Haslam's theories about whether there was a lab leak, who bioengineered this virus, or whether it had a natural origin, aside from all of that, I just want to emphasize that in this conversation, you'll hear a lot of evidence that we do indeed today in 2024, 2025, have the capability to make such viruses, which are very dangerous. And I think that's, aside from your opinion about COVID origins, going forward, thinking about policies to control the possible production of viruses that could lead to new pandemics, that is something that should be in the public discourse, that this is a capability. This kind of genetic engineering is a capability that exists now and needs more scrutiny. I hope you enjoy the episode. Jim, welcome to the podcast.
Thanks for having me, Steve.
So I was explaining to Jim that I have not followed this subject closely. I'm not taking a position in this podcast because I don't have the required expertise to take a position on what really happened. But I did interview Jeffrey Sachs 2 years ago in October 2022. And that was around the time he had finished chairing a Lancet special committee to investigate COVID on behalf of the Lancet, which is a leading British medical scientific journal. And what he told me was that several of the people who were appointed to the committee with him to do this investigation included people who it later turned out had severe conflicts of interest and were not straight with him about those conflicts of interest before participating in the project, and he later removed them from the project. I encourage everybody to go back and watch that interview. One piece of news that we sort of broke in that discussion was about something called the DEFUSE grant proposal, which Jim and I are going to go into great detail about. That is a proposal to DARPA by individuals who might have been involved in the origin of COVID. And that proposal made very, very specific plans to modify, to produce what's called gain of function in what were originally bat viruses that could have led to COVID, the COVID pandemic. That's what we're going to discuss. One of the things that Jeffrey and I discussed, being both very familiar with scientific funding and grant processes, is that at the time what we knew was that the DEFUSE proposal was not funded by DARPA. But both Jeffrey and I knew that researchers often do the research, even if the proposal is not granted. Sometimes they use funds from other sources where they've already done the research before they write the proposal. These kinds of things happen. And that's what Jeffrey Sachs and I discussed 2 years ago. What really got me excited about Jim is he carefully followed the FOIA revelations, the the congressional investigation into COVID origins. And he has put together a number of pieces which are new to me, which are relevant to what happened to the activities which were proposed in that DEFUSE grant proposal. And at the time Jeffrey and I spoke, we didn't know whether those activities were actually carried out. It appears indeed they may have been carried out, funded instead by NIH, by NIAID, which is Fauci's institute within NIH. And I hope I've whetted your appetite for what Jim and I are about to discuss. Jim has spent a lot of time. He's a real expert at this. I'm going to try to slow him down enough that people who are not experts in this subject can follow what his argument is for piecing together what happened. So Jim, apologies for that long introduction. I welcome you to the podcast.
Again, thanks for having me, Steve, and the opportunity to talk about the evidence. I'm going to pick off, I guess, where Jeffrey Sachs stopped on your podcast. He made a kind of a controversial statement saying he heard it was funded. This DARPA DEFUSE grant, this infamous, bid in early 2018, which eerily resembles what emerged in late 2019 in Wuhan. So I guess I'll start off with the first part of the DARPA DEFUSE project. It's DARPA. It's this tiny, successful, nimble bureaucracy, if you want to call it that, within the Pentagon. It's not inside the actual Pentagon building, as best I can tell. It's outside of it, and it's also outside of it, philosophically speaking. They're radical. That's what they are. They date back to World War II, and it was to take on the Russians and Soviets back then. And they have invented some stuff that you use in your phone, GPS, Siri, drones, Agent Orange, machine guns, the whole 9 yards. A lot of these, are dual use technologies. So they continued into obvious, success through the internet era, and they started to evolve in the biological era. There's a book out recently, and she kind of documented this. But in the biological era, they became.
Just interrupt. My audience is very familiar with DARPA.
Oh, okay.
So a lot of,
all right, I'll just go right to the chase then.
Yeah, a lot of physicists, engineers, tech people, they know what DARPA is. So you don't need to introduce DARPA. But one thing you could do is let's talk a little bit about that. Sorry to interrupt you. Let's talk a little bit about the cast of characters. Okay, so who amazingly, and this is something you explained to me, and I double checked since we first spoke. I went and looked at the DEFUSE grant again. So individuals who are named on that proposal include Ralph Baric, Danielle Anderson. Let's talk a little bit about who these people are. So who is Ralph Baric?
He is the coronavirus hunter on the UNC campus in Chapel Hill, North Carolina. He made his name developing this reverse genetic system that could create a coronavirus, a novel coronavirus. He doesn't copy an existing coronavirus. He can create a novel coronavirus. He is one of the few people, I believe, at least in the industry, that can create something from nothing. So he can basically create life in a novel genome never before seen. His reverse genetic system is patented all the way back in 2001 or 2002. It's proven. He started off actually in Fort Detrick in 2003 before UNC had built him a BSL-3 on the campus of Chapel Hill, North Carolina, which is like a 15-minute walk from where Michael Jordan played basketball. So on that campus, he has developed, now it's a $200 million lab. And he is considered to be the expert on coronaviruses.
So Baric, I'm just clarifying things that you said for the audience. So Baric's lab can take a digital file, which is the sequence of a virus, maybe that was genotyped in Wuhan or in Berlin. He can accept that digital file, and he has the ability to synthesize an actual living virus with that particular DNA or RNA sequence. Very uniquely in the world, he can do that. And he can make changes to the sequence that he receives to create a new form or a mutated, different, engineered version of that virus. When it comes to coronaviruses, he is acknowledged, I think, worldwide as the most advanced genetic engineer of coronaviruses. He has a huge laboratory complex at UNC. I was the vice president for research at Michigan State for 8 years. We had to look into various BSL laboratory infrastructure, things like this during my term as VPR. Professors who can command very large grants from agencies like NIH or DARPA have a huge amount of weight on campus because they're bringing millions of dollars, in his case, maybe $100 million or $200 million cumulatively to the campus. So they have a lot of weight. They're kind of the academic superstars on campus. Baric ran this coronavirus research at UNC, world famous, and probably the leading lab in terms of genetic engineering of such viruses in the world. Is that fair, Jim?
That's perfect.
Good. Okay. Continue with Baric, and then we'll talk about Danielle Anderson, who was also on the grant proposal.
So to continue on his engineering capabilities, a lot of people get freaked out when they dive into it. He calls it the no-seum technique. He literally can hide the scars within a genome. He does it the way he cuts it. It literally, hides itself. And when he cuts it, he eliminates the scar from the actual genome itself. And by doing this, a lot of people are thinking, bioweapon. Why do you want to hide your engineering scars? This is not, civilian application. It's only military application. And the brain just starts to go off in crazy different directions. It is, in virology, it is a beautiful thing to be able to hide your engineering scars. You want to, he's an artist, that has perfected his craft over these decades. And because of this, a lot of people get freaked out. But when you're creating a vaccine, it is a genuine moral benefit to have a genome that does not look like it's artificial to the mammalian immune system. So he's trying to hide the engineering scars, not from us lab leakers. He's trying to hide it from the mammal's immune system. If the mammal sees the genome as natural from nature, it will accept it. And then if it accepts it, infects it, it'll create antibodies against it, whether it's a mouse or in this case, a bat. So this is what he's trying to do. He's trying to hide his engineering scars, not from us lab leakers. He's trying to hide it from nature. And by hiding it from nature, the natural mammalian immune system will accept it and produce antibodies against it. So that's a big, one of the many myths that people go off on is they think he's an immoral character. He's a boring father of four. He's about as, clean cut as it gets. I mean, he's pretty idle, he's an up and up.
So to interrupt again, I want to say on this podcast, we're not going to insult anybody. We're not going to make any aspersions or speculations about the motivations of people. We're going to give everyone the benefit of the doubt. So this guy's a serious scientist. I'm sure he's advanced his field of science tremendously. He wouldn't have $200 million of funding over his career, without having done that. So enormous respect to this guy for what he has done. Now, the DEFUSE grant proposal, what was that meant to accomplish? What were they trying to do? And it involved bats, and it involved inoculating bats, Jim. So explain what they were proposing to do.
So this is another great myth that you brought up. Thanks, Steve. And it's that, the DEFUSE proposal has something to do with humans or HIV or I can't even imagine all the other possibilities out there. No, it's about bats. If you go to the cover page of the DEFUSE proposal, it's 75 pages deep, but just go to the cover page. That's enough to get your brain around. That's another file within the file. In the cover page, they say, we will inoculate bats. We will inoculate bats. We will inoculate bats. I believe 3 times, maybe a 4th time. They're talking about wildlife delivery of a vaccine to these bats. And they go into great detail about how they could do this in 75 pages. So the overall arching theme of DARPA DEFUSE was to deliver a vaccine to bats in the wild. And that is the end goal. Why they have been brought in, this group has been brought into this world of biodefense. And it's an EcoHealth bid with the subcontractor of Ralph Baric and a subcontractor of Duke University, including Danielle Anderson, and another subcontractor called the USGS, the United States Geological Service. They look after the caves in North America where bats are kept. So this is a bat program to deliver bat vaccines to bats in the wild.
So let me elaborate on that a little bit and just correct me if I get any of this stuff wrong. So I think Baric has said himself that primarily his funding did not, up until that time, come from DARPA. It was kind of a new thing for him to work with DARPA. He found the DARPA people kind of odd. People in my audience understand, like, the DARPA program managers are kind of cowboys deliberately. They take risks on speculative technologies. They're a little bit like venture capitalists in terms of scientific funding. They're willing to take risks and do slightly far out things, motivated by core needs of the Defense Department. And in this case, this may sound crazy to people. I didn't realize this actually. When I learned this, I thought it would just seem crazy. But the goal was to suppress the development of diseases like viruses for which bats form a deep reservoir. So bats are something like a 1/5 or a 1/4 of all mammalian species. So if you made a list of all types of mammals, different species, the biggest, a very big variety of those, a 1/4 or a 1/5 of all mammalian species are bat species. Okay, so you have these winged mammals. They're mammals like us, so there's a lot of commonality in the base genetic architecture. But they fly all over the place, and they eat insects, and they're fed on by insects like mosquitoes, and they'll bite bigger mammals. So you have a system that is a natural reservoir for dangerous diseases, which can be dangerous to humans, and really ultimately what the Pentagon cares about, dangerous to their soldiers. So in the wake of the original SARS epidemic, which was, I guess, early 2000s, the Pentagon actually investigated the idea that they would inoculate bats, this huge reservoir. They would spread within this reservoir some viruses which would make the bats themselves more resistant to developing these kind of coronaviruses or other things which could ultimately prove dangerous to humans. So believe it or not, we are genetically, our government has paid money to genetically engineer viruses, designing them to spread within the bat population, in this case in Southeast Asia, in order to make our military safer. And I'm not making this up. This is in the language of the DEFUSE proposal. And in particular, the genetic engineering I'm talking about is a modification of something so that it can be transmitted within the bat population. So if you give the virus to one bat and then you let it loose, it will spread the virus to the rest of the bat population and inoculate them against, further developments. Is that fair, Jim? Am I characterizing what?
Oh, that's perfect, Steve. You're a lab leaker and you didn't know it, but yeah, that's welcome to the nerdy, underworld crowd. But but that's it. They want a what do they call it? A scalable solution to this problem.
Right. I'm familiar with this because researchers at MSU work on malaria, and there are various ways to try to suppress malaria by doing things to the mosquito population, genetically engineering things which will cause the mosquitoes not to spread malaria, et cetera, et cetera. So I'm familiar with this at a scientific level. Without even going into the pandemic, if the pandemic had never happened, we could ask the question, wow, are our scientists really genetically engineering viruses so that they will spread among an animal population? Answer is yes, we are doing that. And these are artificially engineered viruses with a goal. to do what we just described. And yes, scientifically, apparently, it's not my field, but apparently this is something that your tax dollars are being spent on. So, okay, let's come back to the DEFUSE proposal. There's a name there. You mentioned the Duke. It's the Duke National University of Singapore Joint Campus, which is in Singapore. And there's a woman who I guess was employed through that academic institution and who did work at a BSL-4 facility in Wuhan. So that woman is Danielle Anderson. So say a little bit about her.
She's my most interesting character in the book. She's a loner like me. She's, a traveler like me. And she's a bit of an alpha, I learned, as the FOIA slowly emerged. She doesn't come across as that because you don't hear much from her. But her story is that she doesn't move. She lives there temporarily in Wuhan, off and on since about 2017. She is the only Westerner in that Eastern lab, though, in 2019. And we only know that because in 2020, she was the Facebook fact checker for lab leak misinformation.
Of course she was. Of course she was. Yes.
She was running to the defense of her Asian colleagues that said, look, they didn't do this, more or less, I'm paraphrasing, but, it's a clean lab. We do good science and we are good people and we didn't do this. And she's, defending her colleagues, she's to her credit. But who's we? There's also this other misnomer that this BSL-4 in Wuhan has got like some sort of PLA, connection. There is no one. Everyone, has looked at this thing, God knows how many hours, has found one PLA scientist in Wuhan before 2020. There's no military connection to this thing. It is built upon the idea of Western collaboration. This is an idea to eliminate the next SARS spillover event. It is a good idea.
So everyone is probably familiar with these maps of Wuhan, which show the Wuhan Institute of Virology. So that was the center of most people's attention. And there's a woman there called the Bat Lady, Dr. Shi, who works there. But that is a BSL-2 facility, and that is not the facility that we're talking about. So if you look at the map, maybe, 10 kilometers away, something like that, 5-10 kilometers away in another part of the city, is this BSL-4 facility. Now, in BSL-4, you're wearing a spacesuit to go in there, okay? And that's a very serious facility. There aren't that many BSL-4 facilities, okay? This woman, Danielle Anderson, worked there. Jim has tracked her using cell phone technology. We'll get to that in a second, and can assert that she was there as late as mid-October of 2019, just before the pandemic started. We'll get to that in a second. But before we get to that, I want to point out that in the DEFUSE proposal, which was not funded, but laid out this whole chain of activities, collect bat viruses, maybe sequence them, send the sequences to Baric. Baric makes various modifications so they become spreadable, transmissible, scalable viruses for the purposes of inoculation within the bat population. And at the end of that chain is Danielle Anderson, who would be injecting the end product, which is a virus, into live animal models in a BSL-4 facility, because it's kind of dangerous, to test whether it really works. And maybe you'd put those bats in a cage with some uninfected bats and see if the uninfected bats then get infected in 24 hours or 96 hours or whatever. So obviously, scientifically, if your goal was to produce this spreadable, transmissible virus within the bat population, this is what you'd have to do. You'd build your own new virus, but you'd have to test it. And Danielle Anderson was the person in the DEFUSE proposal who was going to be doing these activities, okay? And maybe this is the point where we should say that DEFUSE was not funded, but along the way, maybe just in the last 12 months, is that right, Jim? You figured out that actually chunks of that proposal were funded by NIAID, which is Fauci's institute at NIH. So maybe you can elaborate on that.
So it's a brilliant move by Peter Daszak, the president of EcoHealth Alliance, this contractor in New York City who doesn't have a lab, doesn't have a BSL-2, doesn't have, a genome, but they do have, connections, and they have connections all over the world. And those connections extend all the way to Wuhan. And what Peter Daszak did after rejection, and he's rejected, there are meeting minutes where he is pissed off. He is bitching at the staff in New York City. You're idiots. You uploaded late. We look dumb. We just missed out on $14 million. So this is not the kind of guy that sits around and just says, well, I guess I'll just throw away 75 pages of $14 million and not, submit it to the next, potential buyer. So NIAID has a long, relationship with Ralph Baric, has a long relationship with Peter Daszak, has a long relationship with Duke University and Duke University in Singapore. They do not have a long relationship with the United States Geological Service, but they do have a long relationship with Rocky Mountain Lab because they own it, they operate it. So you can see what happens essentially in 2018, in late 2018. There's the project's moving forward technically. I'll come back to the commercial part. I think it's just easier for me to talk technically, and then we'll come back to the commercial part. But technically, this is the paper that Jeffrey Sachs picked up on. And in 2018, late 2018, Ralph Baric has partnered up with the Rocky Mountain Lab in Montana, Hamilton, Montana, and they're going to infect live bats. This sounds right, like what they were planning to do in DARPA DEFUSE. They're moving forward now in not Wuhan, in Montana. So Ralph Baric has now partnered up with this aerosol specialist in a BSL-4 in Hamilton, Montana, named Vincent Munster, and they take what's called the WIV1 genome, which was isolated by the Wuhan Institute of Virology. Again, the Wuhan Institute of Virology can't engineer new genomes like Ralph Baric can, but they can isolate existing genomes. And one of those genomes was WIV1, and they just upload it to the cloud. Baric downloads it, and he can recreate it. He doesn't even need the actual physical sample via plasmids. So Baric takes WIV1, he sends it to Hamilton. This is late 2018 after they've been rejected.
And just to clarify, this is documented. You're not speculating about this, right? So you have actual documentation. So samples were sent from Baric's lab. These are genetically modified, genetically engineered viruses sent to this NIAID lab, which happens to be in Montana, in order that the virus be, I guess, further modified or at least tested to see that it can be spread through the air. Is that correct? Within these animal species? Go ahead.
At this stage, they're just wanting to infect the bats. So the first stage of any bat vaccine program is to infect the bats, to get it to replicate inside that tiny little mammalian immune system. And they fail. The title of it is, WIV1 does not infect Egyptian fruit bats. They're announcing a failure. I guess that's not very common in published papers. Which is
weirdly in biology they don't publish negative results. In physics we publish negative results because they constrain the physical laws. Like this doesn't work, right? Because X. So, but in biology they don't like to publish negative results.
That's what they did here. They publish a negative result and they give you the next step in the paper. And if you look very closely inside this 2018 paper, co-authored by Ralph Baric and Vincent Munster, I've asked both of them to explain this to me. I've asked them for an alibi, and they have not. So I'll go ahead and go into the details. In there, they say we need a Furin cleavage site. They call it intracellular proteases. This is the term Ralph Baric uses when he's trying to get the bat virus into the bat cell. He wants it intracellular. He wants it inside of it. He wants the virus to replicate inside that bat cell. Because WIV1 without the Furin cleavage site did not infect the Egyptian fruit bat. They're proposing that WIV1 with the Furin cleavage site would have infected the Furin cleavage site. WIV1 is just a genome, again, that was ironically, named after the Wuhan Institute of Virology, but it is isolated and created in Chapel Hill. Now it's in Hamilton, Montana, and now it's a wish list of future items that they basically have. And at the very bottom of this paper, they said, therefore, it would be interesting to test this on Chinese horseshoe bats. And there's only one place on the planet, with Chinese horseshoe bats in a BSL-4, and it's Wuhan. So that's the paper that has brought everyone's attention to. Jeffrey Sachs even brought it up on Tucker Carlson and said they wanted to infect Egyptian fruit bats. And Tucker Carlson goes, really? So this is the first step of what's going to happen later.
So we've introduced a new concept for the listeners. You can tell I'm a professor, right? So Furin cleavage site. It's a very short region of DNA, which is necessary so that the virus can enter an animal or human cell and reproduce itself. And it is claimed that the furin cleavage site in actual COVID, SARS-CoV-2, COVID-19, may have been engineered and placed there. What's spooky for people studying the subject is to find a paper in, was it 2018, you said? 2018, in which these guys are saying, hey, we need a furin cleavage site if we want to be able to infect these fruit bats, right? And when they say we need, I think they're saying we need to genetically modify or engineer that site into this virus that we're preparing for bats, right? So that's the spooky thing that got everybody excited. Now, at some point, I want to have a discussion about the nature of the furin cleavage site, which is in the actual SARS-CoV-2 versus maybe other versions of it that have been found in other viruses or that someone might engineer. I don't know, Jim, do you want to have that conversation now? Should we postpone it until a little bit later in the discussion? It's up to you.
No, just be specific with the question, though.
Okay, so if you go online and you start reading about the possibility that SARS-CoV-2 is engineered, one of the responses that you'll read, and I believe Baric sort of says things this way at various times, there's a claim that in the actual furin cleavage site, the amino acids are PRRA, and that is supposed to be evidence that it's not engineered because another variant, I think RRRA, maybe is actually more effective or something. And so therefore, if you were engineering it, why would you do PRRA instead of RRRA? There's a lot of discussion like this online, which I wanted to address because it always comes up. In fact, before I got on this call with you, I was on a research call on a totally different matter with some of my postdocs in theoretical physics. And I mentioned I'm going to talk to this guy, Jim Haslam. And one of my postdocs was pretty familiar with all this stuff. He said, yeah, but it's PRRA. It's, you know what? So if you don't mind, we could go into that because I'm sure it will come up. People are interested in this. I don't really, again, I'm not an expert in this subject. I do not have a deep understanding of this. I'm just raising this point because it comes up in the discussion all the time. So maybe, Jim, if you could address that.
Okay, so yeah, that's a typical, why would they do it this way if they were engineering it? And they is, let's be honest, it's Ralph Baric. He has pre-confessed to wanting to, insert furin cleavage sites into live coronaviruses about 20% different than SARS-1, and SARS-2 is 20% different. So he has pre-confessed to creating this kind of genome. And why is he, using a P was the first question they all used. And I always counter that argument by saying the restriction site is flanked by or the furin cleavage site is flanked by a furin cleavage site, the furin cleavage site is flanked by a restriction enzyme site, I should say, that basically is called BSAX1. And without that restriction site flanking the furin cleavage site, you have to insert the proline in there. It is by, nucleotide, necessity. The biological gods forced Ralph Baric's hands if he's going to use the BSAX1 restriction site to insert the proline to start off this furin cleavage site. It's just, there's no way out of that. But then recently means 2 years ago with a preprint in about, what are we in, January 2025 as we record this. It was December of 2024. A German scientist has gone in there to the records and searched, he's a biological, mathematician and whiz. I don't know how he did this. He told me, but it still doesn't make sense. He searched the database for PRRA and he found it. He found a complete match for this particular furin cleavage site, found in SARS-2, referenced in 2017 at the University of Iowa. It was a paper that they had, what's called a PAT7. I'm not going to get into the details of it because I don't pretend to know all the details of it, but it's just a molecular address that has basically been proven in the literature in 2007 in the University of Iowa to survive a passage after 30 times over in a humanized mouse model. It's stable, to use the term they use. And this piqued Ralph Baric's interest in 2019. In 2019, he publishes a trypsin paper called RXXR. He's focused on, it's a MERS paper. I'll get into why, but it's a MERS. MERS is another coronavirus, just like SARS, found in the Middle East. And he's using this reference of the University of Iowa in 2017 for his 2019 paper. So he is referencing the molecular clone, the precise molecular clone, to use the German scientist, published paper verbiage, the precise molecular clone in 2019. So that PAT7 is essentially a proline followed by 7 amino acids. And it basically forms this molecular address that stabilizes the genome over several passages.
Okay, so I'm not expert enough to judge this, but I believe what you're saying is that the argument that PRRA, when you find that in the furin cleavage amino acids, the argument you often encounter is that this means it was naturally evolved because an engineer would have done it differently. You're saying that that's not a decisive argument, that in fact, Baric wrote papers in 2019 in which he gave that as an example of something that was still perhaps viable. Again, I'm not an expert in this. I just want to state both sides of the argument. Did I do a fair job of that?
Yeah, that's exactly it. I mean, he has referenced the molecular clone found in SARS-2. He is Ralph Baric in 2019. He uploads this paper in October of 2019. So, do your own, timeline on that one when he's working on this thing. So he has a paper trail many times over pointing towards the furin cleavage site and the amino acid sequence and the nucleotide sequence.
Okay. But just to put a little hypertext marker on this, the assertion is that there is no simple argument based just on the molecular structure, molecular genetics of the actual SARS-CoV-2 that could lead you to conclude that, hey, this would be a very crazy way to engineer something. That there's no decisive, you would say there's no decisive argument that establishes that.
Correct.
Okay. Because I think that in every discussion that comes up, right? So I just want to put a marker there. Hopefully more people, more expert even than you and I mean, you, I'm not an expert at all, but hopefully people who really are experts on this subject will go in public and debate this point further and come to some consensus on it. So let's continue in the story. So there are 3 NIH grants from NIAID, which were funded. And what work did they fund?
So to the commercial side, Daszak, mad, lost, I'm going to win. He's a winner. He's got a $20 million a year outfit, he's a name, people pick on him. I did not think much of him. I thought he was a bit of a bozo. He's not. He's sharp. He's good. So what he did is in 2018, along with Ralph Baric working with the Rocky Mountain Lab in October 2018, they all get in a plane. They all, as Ralph Baric, Peter Daszak, Linfa Wong, Danielle Anderson, Xizing Li, they all sit in front of the Wuhan Institute of Virology and they take a picture in October 2018. They're all there. This is the entire DARPA DEFUSE team in October of 2018 in Wuhan. Okay, this project has moved forward. To think otherwise, there's a photographic evidence of this team together in Wuhan. And what they are doing in 2018, October 2018, they are reviving the DARPA DEFUSE project. So Peter Daszak, in his brilliance, took the DARPA DEFUSE grant. And DARPA, as all your listeners, I guess, know, wants 2 pieces to these biological grants. One's called TA1 and one's called TA2. Technical Area 1, Technical Area 2. Technical Area 1, identify the problem. Technical Area 2, do something with it. Technical Area 1 was essentially collect bat samples in bat caves and identify these nasty genomes that have spillover potential. Does that sound familiar? To those in the lab leak debate, it does. It's called the NIAID R01 grant. It's this infamous R01 grant that people have been debating ad nauseum for the past 5 years. And NIAID's defense was that this is not, a precursor to the SARS-2 genome. Yes, we funded some gain-of-function research in Wuhan, but this grant that you're looking at, you lab leakers, is not even close to the SARS-2 genome. It could not even have produced it. This is Tony Fauci's defense. It was molecularly impossible to have created the SARS-2 genome. So that was essentially Fauci's solid defense against funding the creation of SARS-2. This R01 grant by itself is Xizing Li, the Batwoman, in the BSL-2, working with humanized mice. Another, myth that I want to talk about real quick is humanized mice. SARS-CoV-2 is a very contagious agent. It is the most contagious agent in the history of mankind. It does not infect humanized mice. Never in the history of the world has one mouse infected another mouse. It has nothing to do with humanized mice. A mouse did not belch SARS-2 up into the air and whip around the world in a matter of weeks and lock us down. It infects a lot of animals, but it does not infect mice. So I want to make that clear. I think a lot of your listeners may lean lab leak. I'm not going to speak much for them. Maybe they don't. But if you contend that SARS-2 has something to do with Xizing Li's humanized mice, you have to admit it also infects raccoon dogs. It's the same kind of test. You take SARS-2 and you shove 100,000 infectious particles into the nose of a mouse, you're going to get an infection. They did the same with the raccoon dog. These are what's called laboratory infections. These are, essentially not natural. They do not recreate, the natural pathogenesis.
Can I just, so for the audience, there's a distinction to be made between I take the animal and I inject a huge dose of the virus into the animal and maybe the animal gets sick. But if I put the infected animal in a cage with other uninfected animals of the same species, there's a question of whether it can transmit to the other animals. And mice, for example, don't sneeze, so they can't aerosolize stuff around. I don't know anything about raccoon dogs, but I think, are you making that point, Jim, that it's never been shown that raccoon dogs or humanized mice can spread it? I mean, they could be infected if you literally inject it directly into their circulatory system, but they cannot spread it. Is that the point you're trying to make? Okay.
That's exactly it. And you get it. Not a lot of people do. So I'm basically eliminating Xi Jinping's BSL-2 as a suspect, both commercially and biologically.
Okay. So there's 2 points here. One is, I think everyone acknowledges that Xi Jinping did not have the genetic engineering chops to make something like SARS-CoV-2. Possibly only Baric could do it. Okay. So I think that's pretty well acknowledged in what I've read. I think you're making
it's not. So this is the whole chapter in my book. I have to plug it. So, here it is. It's chapter 14, essentially. It's called He Said, She Said. This gets into the weeds really quick, but I'm going to summarize it as best I can. Both Ralph Baric, pre and post pandemic, says the Wuhan Institute of Virology did not copy my technology because I never uploaded my technology to be copied. He's not giving away his $100 million biotech for free to some, Chinese lab on the other side of the world. This guy is competitive, and he's got a competitive edge. He can recreate something from nothing. So Ralph Baric uses cDNA. This is, the Ferrari, I call it, of reverse genetic, engineering of a 30,000 nucleotide, a huge genome in the grand scheme of things. Xi Jinping in the Wuhan Institute of Virology uses BAC, bacteria artificial chromosome. It's basically a used Yugo. Don't get me wrong, her chops are a lot better than mine, than anyone else probably in coronavirus too in the grand scheme of things. But what she can do is copy a genome and basically manipulate it in a very limited way, meaning the spike protein. She can only swap the spike protein. Both she says this and Ralph Baric says this. She cannot engineer the entire 30,000 nucleotide genome. She can only modify spikes, which is about 10 to 12% of the genome. So she cannot create a new backbone. She can copy an existing backbone. Yeah, and that backbone, she can modify it.
Just to elaborate on that, because I think I mentioned this earlier in the podcast, my understanding is, so the height of capability, which a lot of my listeners will understand this, is if I can sequence, I have a virus in my lab, maybe my lab is in China or in Africa or something, I sequence the virus and suddenly it's a little file. It's just some bits. And I email those bits to Baric. He has the capabilities to synthesize de novo a living virus from that information that I sent and also make some non-trivial modifications. And again, I'm not an expert, but my understanding is that is a unique capability that Baric's lab has. And Wuhan Institute of Virology did not have that capability.
Correct.
And so they were primarily, from the viewpoint of the DEFUSE grant, again, correct me if I'm wrong, they were mainly people who would just go out and prospect and get new samples by going into caves, et cetera, or infected, animals, whatever, sequence the virus, but then send those bits to Baric, who could do something, more advanced with that information. And maybe that's not a collective perspective on this, but that's my understanding.
No, that's perfect. And, just to finish off Xi Jinping's engineering capabilities, it always has 7 segments. 7 segments are the way you chop up the genome and deal with it on some sort of, it's 30,000 nucleotide. You can't deal with all that at once. So you have to kind of modify smaller pieces. So she uses a minimum of 7, and SARS-CoV-2 has an infamous 6. So this is the Brutel paper. The Brutel paper, is basically a pretty good, sharp group of guys went in there and found that the SARS-2 genome has these 5 restriction sites, creating 6 segments. And these 6 segments are somewhat evenly spaced over the entire 30,000 nucleotides. And because it's 6 segments, that essentially eliminate the WIB, because they've always used at least 7 to create a genome. This one uses 6, and guess who uses 6? Ralph Baric. So it is essentially, what he uses.
Okay, so now it's October 2018, about 1 year before the pandemic. There's a photo that you can point to. The people in the photo were all the ones who are on the DEFUSE grant. Longtime collaborators, some specialize more on dealing with bats, some specialize on whole-scale genetic engineering of coronaviruses, right? So you've got that whole group of people there in 2018. And I think you're about to say they did get the money to do
so
what...
back to where I started. I get deviated sometimes. Sorry about that. So Peter Daszak lost DARPA DEFUSE May of 2018. So we're now in October of 2018. The whole group, the band's gotten back together. The biological band has gotten back together. We're in Wuhan. We're gonna, crack open some wine. And we're gonna celebrate the resubmittal of DARPA DEFUSE. So he took TA1, Technical Area 1, and he took that to the side, and he took TA2 and he took that to the side. I got to get TA1 funded first. So we did this in 2018 by resubmitting his grant renewal process. This R01 has a 5-year lifespan on it. It started in 2014 for the Wuhan Institute of Virology, 15, 16, 17, 18, 19. And so it's up for renewal in 2019. So in October of 2018, you can see him propose the renewal of this R01 grant for $3.5 million. This is the infamous grant that showed up, that everyone's debated about. And in 2019, they win it in the summer of 2019. So this is now Peter Daszak recycling the TA1, the Technical Area 1, into the R01. How do I know that? You can go into my book. It's kind of hard to describe, verbally here, but I have columns basically at the end of each chapter, and you can see the exact same personnel in each column. Here's the R01, here's the TA1. You can see the same scope of work in the R01 and the TA1. You can see the same, bat collection, the same samples that they want to go after. So this is now the TA1 of DARPA DEFUSE has been funded by this R01 grant starting in 2019. And there's one particular addition to the personnel in this R01 grant that got renewed in 2019 is the addition of Ralph Baric. He was never on this sub-grant to the Wuhan Institute of Virology. He is a high-dollar scientist. What is he doing on this sub-grant to the Wuhan Institute of Virology? I mean, he brings a lot of payroll to the overhead. So this was such a big question. Ralph Baric, why were you added to this R01 grant, this renewal in 2019? It came up during his testimony.
Just to clarify, so he was on the DEFUSE proposal, but he was not on the initial R01 until the renewal happened in 2018. He was added at that point to the renewal. Go on.
So the committee, the COVID committee led by the Republicans, went in there and finally got the guy to talk. And, Dr. Baric, after 3 years of silence after DEFUSE leaked, he decided to, finally give us something. And he did not have a good answer when they brought it up. They asked him point blank, why were you put on this grant? And he goes, I did an invoice. I didn't do anything. But he let it slip that because he's added to the grant, he essentially gets access to Xi Jinping's freezer. That was the entire, purpose of bringing him on to the R01 grant. He now gets access to what Xi's collected for the past decade that has been, properly accounted for, and properly uploaded into NCBI databases, NIH databases. She has gone out there and done her duty. She has been paid to collect these samples. She has been paid to, isolate what she can. These are not dangerous, endeavors, by the way. If someone gets infected, it's just the technician. Mm-hmm. It's not, everyone on the planet. So she has done her duty, and she is also, you can see from the records, she has uploaded or started to upload this bat genome called RATG13 in 2018. This is when she uploads bits and pieces of it to the NCBI database. We could not see it until it leaked, I don't know, leaked is a wrong word, until it was published in 2022. But she is uploading bits and pieces of this RATG13 genome that everyone's debated. It's about a 96% similar genome to SARS-CoV-2 and has made Xi Jinping a primary suspect in a Wuhan lab leak scenario.
Okay, so you've mentioned RATG13. Again, her role in this is, or at least part of her role, or the role in this particular grant, is to go out and get samples, sequence the viruses that are found, upload them to a scientific database, right? So all scientists benefit from being able to search through this database and understand, genetic architecture of viruses and how that architecture is related to what they actually do in terms of infection, et cetera. So it's real science. There's a particular sequence, RATG13, which she had uploaded. And how did that come to people's attention?
Great question.
Yeah.
My favorite question. It's not public record in 2018. It's not public record in 2019, nor is it public record at the beginning of the outbreak. So what happens between, an outbreak in late 2019 and 2020 is she published it. She published it, though, after a lab leak. When she published this RATG13 genome, she basically uploaded it to GISAID. That is a private competitor, I believe, to NCBI or the NIH database, because she can't upload it to the NIH database because she already uploaded it to the NIH database, but it's not published. It's under embargo until 2022. So she went around the embargo. The Batwoman goes around the embargo and uploads it to GISAID, and she punches it into the system on January 24th of 2020. And that is a very key date because I'm going to dive into it here. No one suspected SARS-CoV-2 was engineered from January 10th to January 24th of 2020. I mean, no one. I've seen Kristian Andersen's statements on virological.org. I've seen Bob Gary's statements on virological.org. That's a public record. They're talking about snakes on January 24th of 2020. And they're loving the evolution of this virus as it's emerged in Wuhan because they can trace it in real time through these databases like GISAID. Then she uploads, RATG13 to GISAID on January 24th. Kristian Andersen goes quiet. Kristian Andersen is this evolutionary virologist at the Scripps Institute in California who's got himself into deep water over this whole thing because he's looked at this thing and he does an alignment. He takes Shizhingli's RATG13 and he overlays it with SARS-CoV-2. And I have to do this because it's my visual for everyone. But you see right here is what he's looking at. On January 24th of 2020 is what Kristian Andersen is looking at. So he sees this PRRA, Furin Cleavage site, flanking, this perfect, match across the entire. I wrapped it around the other side of the book just for effects and purposes. It's backwards mirror image. It doesn't matter because it's a 100% match for 500 amino acids, 250 in each direction of that Furin Cleavage site, as if the hand of God, or Ralph Baric, it's separated this genome and put a Furin Cleavage site right in the middle at the S1, S2, and essentially, has just stuck out like a sore thumb because before she uploads RATG13, there's not a great match. This novel genome that's emerged in SARS-2 is so novel, there's not even anything to compare it to. So when she uploads RATG13, she sends out the signal and she says, not my lab. So in my, I'll stop.
I just want to, you're doing great. I just want to make sure the audience doesn't lose track of what's happening here. So let's first not deviate to Kristian Andersen for a second. Let's talk about the motivations of why Dr. Shi did this stuff, and I think why her boss or collaborator, not her boss, but why the guy at Duke resigned. Was it on the same day that he resigned? Okay, so very interesting, right? So at that point, people were pointing to Wuhan, right? So January, people are pointing at Wuhan. Everybody knows there's something going on, right? This is a genome that she had already uploaded to the NIH servers, but there was an embargo. And an embargo typically would be like, oh, I'm going to publish papers on this. So while I'm uploading it into the database, no one else is allowed to see it until typically it would be because we have our patent or we have our papers that are going to come out. And then after that, the genome is available to other labs. Okay. So I'm just recapitulating what you said. If I get it wrong, just interrupt me. So there was an embargo, although she had uploaded it previously. But for some reason, January 24th, 2020, she decides to upload it to this other server. And she makes a public statement, right? Does she? About this, I don't know exactly what she said. Maybe you can fill that in. But
not my lab. Yeah,
not my lab, right? So this thing is not itself dangerous to humans, right? RATG13. It's just a thing found in the wild. It's a bat virus. But then later on, people find that aside from this Furin cleavage region, there's a very, very close match between RATG13 and the actual SARS-CoV-2 virus. Do I have all that right?
That's perfect.
And at the same time, the Duke professor, who is Danielle Andersen's boss, okay, a bat guy, a coronavirus guy. He resigns, right? They were the ones with access to that BSL-4. She was at a BSL-2 across town, but the fingers were all pointing to her. Okay. So
ironically, the fingers pointed, though, to she for the past 5 years because she brought attention to her lab by publishing RATG13. It made her the primary suspect. It backfired, in other words.
But I think your interpretation and her own statements subsequently have been, look, I didn't have the ability to engineer this. We did find RATG13, but RATG13 is at best a precursor, or at least it's similar to the actual dangerous SARS-CoV-2, but it doesn't have that Furin cleavage site.
Correct. And the counter to your argument, you often hear from lab leakers. I'm a second generation lab leaker. I love that term. I just piggybacked off the work of first generation. But their argument is that she took the Wuhan Institute of Virology took their database online, and I counter that by saying it's biologically impossible to publish something closer than RATG13. When you look at a 500 amino acid match over, this very important S1 S2 junction, and there's a Furin cleavage site in the middle. I call it, in the book, I call you naive. In the Substack, I have a more, graphic word to use. You're not really thinking this through. I mean, by publishing this natural genome, she made the SARS-2 genome look unnatural.
Okay. Before we get into that, because now we get into what Kristian Andersen noticed right away when he aligned these two genomes and saw this Furin cleavage site stand out. Online, I see people talking about Banal 52. And they say, Banal 52 is actually more similar to SARS-CoV-2 or something like this. It's not actually RATG13. So maybe you can just comment on that.
Oh, it's like everything in lab leak. It's another, endless rabbit hole. But Banal stands for bat anal. So that just cuts to the graphic side of this thing. And it emerged well after the pandemic started in the September of 2021, about the same time Major Murphy leaked DARPA DEFUSE. It was like a 1-2 punch. And this genome was published by the French who have a lab there in Laos. And it's called Laos Banal 52. And I believe RATG13 is like 96.1% identical to SARS-CoV-2. Laos Banal is like 96.8. It's in the same region. But there's a quirk. There's a curveball in this thing. It was also collected by the U.S. military. At least the funds of the U.S. military were in that same bat cave at that same GPS. This Laos Banal 52 sample was found in 2017. It's this, I've already forgot. NMRCA. It's another acronym basically, but the military, again, has a big footprint around, the specific rim. And they're looking, for these pathogens. And this pathogen was found in this bat cave in 2017. And we don't know where it went or where it is. It's also been embargoed somewhere.
Okay, quick question. So that's, I didn't know this. So the bat anal thing beat Banal 52. Do you know which scientists actually found it and sequenced it? So you said it was collected in 2017. So that's even earlier, right? But do we know who these people are? And they didn't publish it, or what?
Yeah, we know who he is, and people have contacted him, and he said it's embargoed. The samples from 2017 are embargoed by the funder, and the funder is the DoD. It is the Pentagon. They did not allow this genome to be published by the French.
So somebody, not DARPA, but DoD, paid some French guys, or were people working with the French guys, and that's how Banal 52 was discovered. Is that
the exact same GPS down to, the fourth digit is, being collected in 2017 and embargoed in a freezer somewhere in Laos. And it's quite the story, and another rabbit hole into itself. But yes, and that the same program manager on that job was even on the DARPA DEFUSE, bids, and it's a fun, they're all the same, group.
Well, okay, just to clarify, so the program, you do know how that collection was and sequencing was funded. It was through DARPA and the same program manager.
It was a guy that was a part of the DARPA DEFUSE type of bids in 2018.
Someone in DARPA who had the infectious disease portfolio. Yeah. Okay, let's come back to Kristian Andersen. Kristian Andersen, originally from Denmark, an evolutionary virologist, so interested in the evolution of viruses. Scripps Institute in San Diego, beautiful office overlooking the Pacific Ocean. January 24, or maybe a few days after, I don't know exactly when, But after she publishes or releases the RATG13 sequence, he's looking at it. And by that time, they also have some early SARS-CoV-2 sequences, right? And he aligns them. Okay. So when you align them, you can compare base pair by base pair, the two sequences. And what he found was something that stood out very strongly in that there was enormous similarity over most of the two genomes, but it looked like this Furin cleavage site had just been inserted. Like there was a break and someone had inserted this Furin cleavage region. Kristian Andersen, immediately alarm bells went off in his head. We have his emails and Slack conversations with his colleagues who are also evolutionary virologists. And then later, people like Fauci and Jeremy Farrar were pulled in. But I want you to tell that story.
That's my favorite part of the story. It's chapter 15 of the book. It's on the Substack. You know, it's when it starts to rock and roll. So again, Kristian Andersen on public record in virological.org, where all these evolutionary virologists hang out, is on record saying this is beautiful genome from nature, snakes. He is full on board with the natural spillover event and excited. And then Shizingli uploads RATG13 on January 24th. And you can actually see one of the virologists upload the paper into the forum. And then all of a sudden underneath that forum, it goes quiet. There's no more Kristian Andersen. There's no more Bob Garrett. There's no more anyone in there. And now we know what happened. They went to, burner phones. They went to Slack messages. They went to, holy crap, this thing is engineered. And Kristian Andersen is ringing he's paranoid. And he's also ringing the biological fire alarm. He is by January 30th of 2020, he wants to call the FBI. He testified that he had FBI and CIA pamphlets ready to go. He has not talked to Fauci. He has not talked to anyone besides Bob Gary and Eddie Holmes in Australia. And I think when he talks to Eddie Holmes in Australia and he shares his, concerns, because he's like, hey, Eddie, have you looked at this? He goes, yeah, I looked at the SARS-2 genome. There's nothing there. As far as I can tell, it's natural. Then, Kristian Andersen brings the attention to the RATG13 genome to expose the SARS-2 Furin cleavage site. And, I think, Kristian Andersen said, I wanted to pound some beers after that call with him. Well, Eddie Holmes gets on the call and calls us handler. He calls him, it's Jerry Farrar, the Wellcome Trust. So Jerry Farrar, the Wellcome Trust, is now pulled into this thing. He says he heard some rumors from some, U.S. virologists. It's code for, Kristian Andersen, because he's the only one on record basically going out there to ring the biological fire alarm on SARS-CoV-2 being engineered. It was before the February 1st teleconference. This is all before the February 1st teleconference. There's nothing to do with the February 1st teleconference. So I, looked at all these emails they have, and it's not easy to figure out what's going on. But Jerry Farrar, the Wellcome Trust, the Fauci of the UK, has received these concerns directly from Kristian Andersen. And he's actually genuinely concerned, as best I can tell. This isn't like some, he's going to help cover this thing up. He is genuinely concerned. Like, wow, this evolutionary virologist, this world-class evolutionary virologist with a corner office overlooking the Pacific Ocean, bringing in his own, grants, is going to call the FBI. Let's talk about this. It sounds like a logical thing to do. And he is organizing this February 1st call with Ron Fouchier, with Marion Koopmans, with Christian Drosten. These are all the Faucis of like Denmark, and not Denmark, the Netherlands and Germany. These are the who's who. He's organizing this, and Fauci's not involved at first. And he keeps sending Fauci emails, and only Fauci in the U.S. government, hey, we're doing a teleconference. One of your own virologist in California is ready to call the FBI. And Farrar, to his defense, has documented this in his book, Spike. Am I supposed to call the FBI? Is the actual quote from Kristian Andersen into Farrar's book on chapter 3. That's the name of the chapter. Am I supposed to call the FBI? And he puts on there January 30th. It's almost like he's, covering his own hide. And I don't really blame him. He doesn't get pulled out of this thing any further than he has. So he's begging Fauci to get on this phone call that's been organized on January 31st. And that night, January 31st, Fauci finally picks up the phone and calls Kristian Andersen. And Kristian Andersen, we don't know what's said, but apparently Kristian Andersen has relayed to Fauci, I'm ready to call the FBI. This thing's engineered. And then, they hang up the phone And then the email comes from Kristian Andersen, almost as if he's trying to like cover his own hide on this thing. He wants to document what he's told Fauci. We find the genome inconsistent with expectations from evolutionary theory. We is Kristian Andersen, Bob Gary, Andrew Rambo, and Mike, I forget his last name. But these are 4 world-class evolutionary virologists, hang out in your circles, not mine, going to Harvard, Yale, and all that. One of them, Eddie Holmes, they're all world-class virologists. And they have basically told Tony Fauci on the night of January 31st, 2020, we find the genome inconsistent with expectations of engineer. And he says that you have to look very closely at the genome to see that it's engineered because there's very small pieces of it that looks potentially engineered. And he's talking about the Furin cleavage site, essentially, at that point. It's pretty obvious he's admitted that. That's January 31st.
So what we've just described is the first alarm bells going off once they had the RATG13 genome and they could compare it to the by then sequenced SARS-CoV-2 genome. People who were completely outside this circle of coronavirus gene editors and bat scientists, people who were completely outside, who were well-respected evolutionary virologists, got alarmed, started talking about it amongst themselves, pulled in Jeremy Farrar, who is the head of the Wellcome Trust, which is the largest funder, I believe, of biomedical research in the UK. I actually know Jeremy. Coincidentally, I was visiting Jeremy in mid-January 2020, so about 2 weeks maybe before these events that you're talking about happened. It turns out around Jeremy, and this is documented to some extent in his book, Spike, which I highly recommend, his lieutenant, deputy at the Wellcome Trust, I think is a former MI5 or MI6 director, and one of the members of his board was also had a very senior position or maybe top position at MI5 or MI6. So he's surrounded by spooks, which I found very strange. In his book, he says once the shit hit the fan, once this broke, the events you're talking about, he notes that he was advised immediately to get a burner phone and a separate email account. And I always found that very strange because most scientists don't get burner phones ever. Okay. So, but now maybe it's just because he was surrounded by spooks and there was, as the British might say, an abundance of caution. Okay. But he writes about this in his book because he also found it strange. Okay. Now, having said all that, which makes this all sound very exciting and James Bond-like, I just want the listeners to take a step back and we are talking about what people at a moment in time thought. It is possible that they were mistaken and later changed their minds. So I want to allow for that possibility that Kristian Andersen, others were very alarmed, rightfully so, had this set of conversations. However, they later wrote a paper called Proximal Origins, in which they advocate for a zoonotic or natural origin of the virus and against human engineering of the virus. We'll get to that discussion in a few moments. But as of right now, it is February 1. There is a conference call involving scientists from all over the world. Kristian Andersen, the guy at Scripps. I think Fauci is on this call. Farrar is on this call. And Jim has learned, or I learned from Jim, that secretly, without anybody else's understanding, except maybe I think Fauci, who invited him, Ralph Baric is on this call. Am I right about this? February 1, 2020. Go ahead.
February 1. So it was always a question. Why did these world-class evolutionary virologists go from I'm calling the FBI to I'm going to write a paper saying this thing came from nature in a matter of, days, weeks, months. And it never really made sense ever. And everyone thought they, did this through, the scientific collaboration and feedback and peer review system. And they got talked down from their conspiracy. They called it, this thing was engineered. So it never really made sense to anyone, including myself, why on earth this group, did a 180. And the assumption was that someone like Kristian Andersen was bribed maybe to change his answer. And that assumption was based on this proposal called CREED. I don't want to get too far off track, but essentially that's part of the DARPA DEFUSE also funding. So that was always the Richard Ebright of Rutgers University called a creed pro quo. So that's the narrative. He changed, Kristian Andersen changed his mind on engineering because of a bribe. And bribe is the word that Fauci used.
I just want to be very clear that we're not making any assertions. You're stating that some people have expressed this online, for example. I actually know Richard. We're not asserting that here. Okay, so let's leave that aside. But it is true that people who were very alarmed and caused this teleconference to happen on February 1 later are authors of a paper claiming it was not engineered. But let's go through it. So, and what's interesting now, thanks to Jim, is that we have, thanks to all the hearings and the FOIAs, we have access to the communications of these people in the days when this conference is happening and the transition afterwards in their opinions. So go ahead, Jim.
So Kristian Andersen's files that he used are presentation slides, PowerPoint presentation slides that he used during the February 1st teleconference are public record. And the big one and the main one is the genomic alignment of RATG13 over SARS-2. And it's essentially what we've already talked about. I can now point to this Furin cleavage site in the middle of this genome that's almost been inserted out of frame, a perfect, S1, S2 junction. This is not nature. This is man. And that's essentially what's going on February 1st. And they hang up the call after about an hour. And on the public record, only showed Fauci and Collins had invited Tabak. And so there's 3 U.S. government officials, and that's it. Everyone else is, not works for the U.S. government. They're a spook like Farrar, or so, it's someone else, and somewhere else. So that's what happens on February 1st. It's a gray area. Then comes this follow-up meeting. This is now organized. This thing's been escalated. To Fauci's defense, he organized it. He says, I'm going to get a broader group of virologists together, and we're going to do it under the NASM umbrella. The National Academy of Science has basically, on February 3rd, this is now a Monday, February 3rd of 2020, has gotten together to expand upon Kristian Andersen's concerns of engineering. And you can see Kristian Andersen go into this meeting kind of like cynical. NASM's a political organization. They don't understand the genomic evidence, but I'm just logging off here, guys, and I'm going on.
And when you say he was cynical going in, we know this because his emails and Slack messages to his close collaborators expressed these sentiments before he went to the NASM meeting. Just to be
correct.
Yeah.
His words, not mine. And he logs off, the Slack messages, and he goes into the NASM meeting. And now this meeting formally includes Ralph Baric. And we did not know what occurred during this meeting. We knew less about this February 3rd meeting. This is now U.S. government. This is now FBI. This is now CIA. This now is the White House. This is the who's who, basically. We now have gone from a bunch of foreigners and virologists debating it to U.S. government officials are now debating the genomic origins of SARS-CoV-2. And during this meeting, Kristian Andersen represents the exact same, slides. You can see him working on it. He's such a brilliant nerd. He's working on this.
Y'all are.
Okay. He's working on this proof that I'm going to prove this thing's engineered, and I'm going to do my patriotic duty, and I'm just basically going to be, everyone's going to love me. And, he represents his slides to, as far as I can tell, they're the same ones. And again, it's the genomic evidence showing RATG13 over SARS-2 exposing this Furin cleavage site. Kristian Andersen's words were redacted. Once he came back into the Slack forum, his reaction to the February 3rd meeting organized by NASM, they were redacted by Kristian Andersen's own lawyers. They did not want it to be public record. This was so well hidden, no one saw what happened during the February 3rd meeting. But to use Kristian Andersen,
on what grounds can they redact? I mean, weren't these? How are these Slack messages extracted? I mean, under what?
There was a negotiation between the congressional committee.
Got it. All right.
Yeah. And, we'll give you the Slack messages, but we're not going to give you everything. And, we're going to give you the least incriminating bits and pieces. So one of the congressional committee guys, I know which one, I don't want to say him, he marched down to the HHS office, which is in downtown DC. And he went into what's called an in-camera view, I believe they call it, where you go in and you can't take a picture, but you can take notes about what's actually behind the redactions. And he went in there and he pretty awesome truth seeker. He wrote down what happened after that February 3rd meeting, and he brought that into the Ralph Baric testimony. And he says, look, I've got something to read to you, because under Kristian Andersen's testimony, they invited everyone but you to that February 1st teleconference. And they wanted to make sure you weren't there because they knew that, this is Kristian Andersen to the committee in 2023 during his deposition. They wanted to make sure that they had, an objective viewpoint, because they knew Ralph Baric had been collaborating with the Wuhan Institute of Virology. There's an infamous 2015 paper, real quick. The only reason Xi Jinping's on the paper is because Xi Jinping shared the sample before publication, so Ralph Baric would add her as a co-author. This is something that similar happened with RATG 13, obviously. She's only shared the sample before publication, and she'll be added as a co-author to a Nature paper. But anyways, they know that Ralph Baric collaborates, so they did not want him on the February 1st teleconference. But what Ralph Baric did is listen to Kristian Andersen's evidence secretly on the phone, not visually, and attacked is the word Kristian Andersen used. Ralph Baric just attacked me. I wonder if he's implicated in this too. So Ralph Baric attacks Kristian Andersen for suggesting his SARS-2 genome, this SARS-2 genome looks potentially engineered.
Okay, so just to recap, these were recovered by some congressional investigator who took hand notes, being able to read the unredacted Slack messages. So upon leaving this NASA meeting, Kristian Andersen says to his close colleagues, I was attacked by Baric in this meeting, who potentially is implicated in all this. Okay, go on.
And so that's the 180. It was always a question in my mind, why on earth did Kristian Andersen, a pretty cocky guy by anyone's account, a pretty confident guy is the word I should use, why did he 180? And, I'm not going to get into the psychological aspects of this, but if you're the Corona King, and you're, attacking someone that's an Ebola specialist, who's going to win the argument, And he did it. Ralph Baric did it in front of the CIA, in front of the FBI, in front of the White House. When everyone was watching, he attacked them as if, I'm the alpha of the room and this thing's not engineered. And so, Kristian Andersen, I don't blame him. I would have, folded. I mean, maybe your evidence isn't all there. He's getting gaslit by the king.
So just to, again, for the audience, Baric is the expert on genetically engineering coronavirus. As far as I know, Kristian Andersen is not a genetic engineer, right? He's not a guy who goes in the lab and actually develops techniques for modifying genomes, et cetera. So if I went to that meeting and I were from a more theoretical viewpoint, like an evolutionary virologist, and I study sequences and I align them and I look at how they change over time, and I do things like that. But somebody who's an engineer comes in and says, hey, this is natural because we would never do it this way. We would, et cetera, et cetera. Just to give Kristian the benefit of the doubt, I could imagine him just saying, well, somebody with deeper knowledge about engineering is telling me it's natural, it's not engineered, this is not how we would do it, and maybe I just accept that. So we don't have to talk about bribes or, it's possible, okay? I just want to lay that out. It's possible he's got the highest level of intellectual integrity, and he was just convinced by a stronger area expert about this conclusion, right? Why would you not believe that?
Oh, I would fold in a heartbeat, there's an evolutionary gene in us to go along to get along.
Right. But okay, but let me go forward now. So first of all, are you sure he folded right away? Like maybe for a week or some period of time, he's still thinking about it? Or how do you know that was the key thing that changed his mind about it?
Well, I'm not going to get into his mind. He has communicated with. I'm not going to say that. I don't want to talk about that offline. But I will say this. It's a public record question. Did you know Ralph Baric, Kristian Andersen? Did you know Ralph Baric was on the February 1st teleconference? No, I did not, and we were shocked to learn that. So he had no idea Ralph Baric was listening in to his February 1st.
Yeah,
evidence.
no. I don't question this idea that he was ambushed. And by the way, that's a pretty dirty trick on the part of Fauci or Collins or whoever invited Baric onto that call. It's a pretty dirty trick, right? I have no doubt that he was ambushed at this later meeting.
Ambushed is the word.
I'm more interested in, did these people who originally saw this smoking gun kind of thing and thought, oh, hey, this is engineered, but then became authors of this paper, which came out a few months later, I think. At what point did they change their mind? What in the record speaks to why they changed their mind? Because the paper itself, I found, which I read at the time when it came out, is very unsatisfying. So I'm now interested in this time evolution between February 3 and when that paper came out and any insights you can offer about that.
So that's chapter 15 in the book. I have direct quotes from their emails in there. You can actually see them kind of convincing themselves, to be honest with you, maybe this thing is not intentionally engineered. If you read closely, what they're going to argue is that this thing, it could have been passaged in an animal. Maybe the Wuhan Institute of Virology messed up and it got out, but it was unintentionally engineered to, have a Furin cleavage site. But if there's a point to the proximal origins paper, it is to make a point that it's not intentionally engineered. And my point is that it was intentionally engineered. This is actually, the intent of this genome was to infect an animal, not us humans.
Okay, so the particular point about that particular group of authors of proximal origins, what went on in their heads, maybe it is a little secondary. Let's get to your main thesis, which is that going all the way back to the original DEFUSE proposal, which DARPA asked for, right? We want to be able to create a virus which transmits within the bat population. It's not aimed at humans. It's aimed at transmitting within the bat population, but that will maybe reduce the dangerous reservoir of diseases which are lurking in the bat reservoir, which could eventually affect humans. But initially, we're just trying to affect what's going on in the bat world. Your contention is that this process was followed, and the most plausible thing is that ultimately they did engineer that Furin cleavage site into something which might have been similar to RATG13. That was then further processed at the Rocky Mountain Lab. You have records of things being sent to the Rocky Mountain Lab, which is an intramural internal lab of NIAID. And your further contention is that eventually that virus was sent to Kristian Andersen in the BSL-4 in Wuhan. And that is
Danielle Anderson, yes.
Danielle Anderson. And that is really the origin of SARS-CoV-2. So we're finally at your main, sorry it took so long, but I want the audience to come along. That is I think your main conclusion. When you write mystery solved on the title of your book, that is what your contention is. So let's just talk us through that.
So where do I start with her? I got to make it abundantly clear. She's what I call the first victim of many. I start the book off, though, with her. And by naming her, as patient zero, it is to help the reader move along to the end of the book. She is a character to deliver, a narrative that delivers an answer. And the whole premise about her presence in Wuhan has never been answered. And she was always kind of in, the background. Who is she? What is she? She's a world-class bat immunologist. I turned myself into an amateur bat immunologist to try and figure out who she was. She's not easy to understand. She has been asked by her own colleagues to explain what she does and what she was doing inside that Wuhan lab. And you can listen to the presentation. It's on YouTube, and it's not even easy to follow because it's very complex. But when you're a bat immunologist, which is what she is Lympha Wong Jr. She is Lympha Wong. Lympha Wong is the Batman, and she is, the Bat Lady, we'll call her. She's more of a Bat woman than Xi Zhengli.
Lympha Wong was, in a sense, her superior was the Duke NUS, National University of Singapore, Duke campus in Singapore professor. And he's the one who resigned his position on January 24th when...
January 10th, the born-on date for the SARS-2 genome.
Okay, sorry.
He resigns on January 10th. He resigns as Duke's emerging infectious disease director to spend more time with the family.
Okay.
But January 10th is
I just
the date.
Because you mentioned his name, so I just thought I'd explain to the audience who that is. Danielle, I guess, worked under him, and she was really the one in the lab. I mean, you've got photos of her with her BSL-4 spacesuit on in the lab, and you've traced her physically to that BSL-4 facility in the lead up to the actual pandemic. So go on with that, and also maybe tell us how you traced her.
So there's an NBC cell phone news report that was released in May of 2020, and it says there's an event inside the Wuhan Institute of Virology, inside the BSL-4, in like the first full week of October of 2019. And inside that NBC News report is a link to the raw report. And inside the raw report is about 15 pages of the data. And, everyone can go look at it themselves. NBC News cell phone report, and just click on the link inside the report and you look at the raw report. If you open up the raw report, you can come to your own conclusions about what's happening. So inside this raw report are 2 cell phones that they've traced. There's a cell phone that bases in Singapore, and they literally declare is the emerging infectious disease director, but the name's redacted. And it's one of the 2.
That's Linfa.
That's Lympha Wong.
Yeah.
But the name's redacted, so, but by inference, who they've told you, it's him. Then there's another report. There's another page that's dedicated to another cell phone. And this one's the world traveler. This one is getting around. This one is all over the place. It starts off in Kenya in 2018-19, where there's a Duke hospital. And then there's a Danielle Anderson publication on MERS and camels there. Then her phone, this phone that's also had its name redacted, goes over to Beijing in the early of 2019. Now she's working on a CRISPR paper inside of a Beijing lab, inside of a BSL-3. She's the lead author on that publication paper in 2019. She is international. She's a Western woman from Australia, but she is truly international. And she's, as best I can tell, invited in country into China under the Chinese Academy of Science. She is, working, to prevent the next spillover event, essentially.
None of this, for scientists, none of this is surprising. This is the life of a scientist. So I gave a talk at the Chinese Academy of Sciences like a month and a half ago. So this is an international world. I thought oil and gas was international. This is on another level. I mean, she describes her passport as being like filled up 3 times over. Yeah, I've had to use one up and get a new one actually. But wow. Okay, but most importantly, cell phone logs place her both at the BSL-4 facility, and then there's a dormitory where she sleeps in Wuhan, right?
Correct. I don't have her cell phone at the dormitory per se. I have her word of the dormitory, but then she flies from Beijing. This phone flies from Beijing to Wuhan on June 28, 2019. That is the application date for the CREED project. This is the CREED program that's been called the CREED Pro Quo, the bribe to Kristian Andersen. Kristian Andersen, in his defense, was true. He said this thing was in the works for a long time, and it was in the works. The bid was due on June 28, 2019. This is the CREED project. This is now Peter Daszak has taken TA2, Technical Area 2, and repackaged it into this CREED grant, essentially. And only now have we recently got the FOIA to see what's going on there. But June 28, 2019, Danielle enters the BSL-4 after giving input into the CREED grant. She is actually a bit of an alpha. I did not know this until I read her input into this CREED grant. She is steering Peter Daszak, and she is steering Ralph Baric away from a coronavirus-centric bid into a more general bid. This is her pushback and feedback from them losing DARPA DEFUSE. She's mad they lost DARPA DEFUSE. She wants, to have a big grant to work on.
Yeah, and I want to defend all these people because in their minds, in her mind, there probably was a good rationale to pursue all of this science. It's real science. How do these viruses work? How do they get into the human body? How do they divide? How do they get into your cells? How are they transmit? This is all real science that we need to understand. And if the military says we want a scalable, transmissible thing, which is going to improve the situation with the bat disease reservoir, and we're funding it, most scientists are not going to say no to that, right? They didn't know this could potentially lead to a pandemic, et cetera. So I don't want to paint a negative picture of these people. They're doing real science. They're traveling around the world doing it. But it may have led to something, and that's your contention. Yeah.
Correct. So she gives input to the CREED bid, which is Technical Area 2 of DARPA DEFUSE. It has basically the same scope of work. Danielle Anderson's going to head the animal challenge studies. Ralph Baric's going to do the genomic engineering. And inside of this CREED bid is a draft that they're going to submit. And inside the draft of this CREED bid in 2019 is Ralph Baric's Furin Cleavage site, a reference to the Furin Cleavage site. It's the exact same reference he used in DARPA DEFUSE. So Ralph Baric has taken his proposal from 2018 and refurbished it. And now he gets to try out his novel new genome at the Furin Cleavage site in this CREED grant that's about to be funded by NIAID. So this is not in the final grant. This is in the draft. Okay, it did not make the draft because I'm guessing DARPA allows 75 pages and NIAID only allows like 15 or 25. That was the beauty of DARPA DEFUSE. We got 75 pages of their mindset in early 2018, but it's shrink down to make it fit. And Danielle's making it even broader and less SARS-centric as they go into their research of what's about to happen.
Okay, but I want to back up a little bit because now you're already in mid to fall 2019 when she flies from Beijing to Wuhan.
June 28th or the end of June 2019, and then she's in the BSL-4 until October of 2019.
Right, but I think there's a part of the story that you want to tell, which is that Baric sent some sample viruses to the Rocky Mountain lab, which is an intramural NIAID lab in Montana. And what was done there, and what was done there was then, you contend, sent to Danielle Anderson, and that's what she was working with in that BSL-4, you contend, right? So go ahead with that.
Yeah, that's a lot to digest, but there is a record, a FOIA of Ralph Baric working on an RRAR Furin Cleavage site and shipping, MERS-like samples to Rocky Mountain Lab in February of 2019. It's documented in the book. It's public record. You can ask me for the references on that one. It is essentially, I'm now working with Rocky Mountain Lab, Rocky Mountain Lab's going to do the in vivo experimentation of my in vitro genome. I am now, before the pandemic, Ralph had a great line. I never sent Chimera to China, and that was always his defense, and it was a pretty solid one. And that's before DARPA DEFUSE leaked. That was like, hey, I never sent Chimera to China. And that's when my mind perked up. That's an interview he gave to MIT Technical Review in like June, not even that, maybe May or June of 2021, a few months before DARPA DEFUSE leaked, then he went quiet. But I never sent Chimera to China. That always piqued my interest. Who's the third-party lab? And I found that 2018 paper where they're collaborating together. So Ralph Baric has a FOIA that says, I'm going to, the in vivo experimentation on this genome I've created is going to be done in Rocky Mountain Lab. And that's February 2019. So in there, obviously, this 2018 paper where they tried to infect the Egyptian fruit bat, is now carried into 2019. And if you look deep into the Creed grant, the Rocky Mountain Lab is a part of it. This guy, Vincent Munster, is a part of it. So they have now gotten the DARPA DEFUSE team back together with the DARPA DEFUSE team that won. The DARPA preempt team that won was in Rocky Mountain Lab. They beat, Peter Daszak and Ralph Baric in 2018 with better technology. They have what's called transmissible technology. Baric's technology was transferable in DARPA DEFUSE. So Vincent Munster and Rocky Mountain Lab have transmissible technology. That's inject the needle into one animal and it spreads to the next animal via aerosols. This is highly dangerous as far as I'm concerned because it's an infinite loop that you're potentially creating. But inside of a BSL-4, they thought they were safe and sound. So this bat vaccine has been developed inside Rocky Mountain Lab using what's called an Egyptian fruit bat.
So your contention is that the further processing of what Baric produced was done at the Rocky Mountain Lab. For sure, there was some relationship where testing it in vivo in the actual animals was going to be done for some things that Baric produced in that lab. I think there's records to that effect. But you can't directly establish that something was sent from that Rocky Mountain Lab to Wuhan BSL-4 where Danielle Anderson worked. Right. So you assert that's true, but you don't have the strongest evidence for that, right?
Correct. What I have is the return label. So the whole book, the whole genesis of the project I took on was to answer a question. Why does this old world virus called SARS-CoV-2 love new world lab animals? That's it. That's all I care about. Why does SARS-2 from the old world, the Eastern Hemisphere, I think we all agree to that, love new world lab animals? And by new world lab animals, I mean, Western Hemisphere, animals that you find in a BSL-4 lab. One of them's Egyptian fruit bats. It's called the lab rat, basically, of BSL-4s in Western countries. It's easy to feed and breed. It's easy to, keep up because it eats fruit. You just throw some fruit into the cage and the bat will eat it itself. The Chinese horseshoe bat is where the test needs to be done. This is actually a high maintenance bat. It's an insectivirus bat. It basically eats, flies. And it's very high maintenance to keep up. So if you're going to test a bat vaccine that you've developed in Egyptian fruit bats, you have to go to Wuhan to test it. And the only place on the planet, basically with live Chinese horseshoe bats. Danielle Anderson has admitted she had Chinese horseshoe bats inside the BSL-4. to Peter Daszak, who admitted she was working on a live bat colony. She claims the bat colony didn't work. It didn't, it failed. The word colony, though, doesn't mean much when it comes to Chinese horseshoe bats, because what they proposed in DARPA DEFUSE was wild caught Chinese horseshoe bats would be kept inside this Wuhan BSL-4. Wild caught means I went out to the wild, I grabbed a Chinese horseshoe bat, I brought it into the lab, and I'm going to keep it alive long enough for experimentation. It's not born into captivity. It's not bred, ideally is the way it's supposed to be done. So it's a bit of a, it was in the DARPA DEFUSE documents that, hey, we can get the WIV colleagues of ours to basically catch wild-caught bats. So she has admitted, to Peter Daszak, who admitted publicly, ironically, weirdly enough, that she was working on creating a live bat colony, and it failed. So that is her admitting live bats are in this BSL-4.
Okay.
When it hit the fan, she has a story that's different than what she was working on. She claims she was working on Ebola. So I've gone into her CV. It's 100 pages long. It's 100 papers long, and I've looked for it, and I did not find one paper on Ebola. I asked her colleague at Duke at the time, did she ever work on Ebola? He didn't, he kind of said no in a professional way. And I showed him the quote in the Bloomberg article, well, she claims she was working on Ebola, so that's not adding up. She never disclosed that she was on the DARPA DEFUSE team. So her story, basically, post-pandemic, is not adding up.
Okay. But you mentioned another line of evidence, which is the New World animals. So let me summarize what I think I understand about that, but please correct me. So I think your claim is that the actual SARS-CoV-2 virus is transmissible within certain other mammalian species. And they include, I think, deer mice, is it mink or ferrets, things like this? I don't know if cats or hamsters are in there, but it's some very finite set of species. And there is some link between those specific, I think, 5 species and what they have at the Rocky Mountain lab, right? This is an important part in your chain of logic. So could you go into that?
So that's it. There's 5 animals on the planet outside of us humans that SARS-CoV-2 efficiently transmits through. Onward transmission is the term I've come across. That means I can infect you and you can infect Bob and Bob can infect Kay and Kay can infect Joe. It's onward transmission. Cats are a dead end. Us humans can infect the cats in captivity, in the house, but they don't infect other cats. There's no genomic evidence in GIS-AID of many cats being infected. There's no genomic evidence of raccoon dogs being, infected multiple times over. There's no laboratory evidence of humanized mice being infected. It was a big problem at the beginning of the pandemic. What does SARS-CoV-2 infect and, more importantly, transmit? Because we need to create antibodies, vaccines to stop the transmission. It's always been the problem to replicate transmission of SARS-CoV-2. And over time, this pandemic, has created, its own laboratory. And we found out what we, essentially is, people have published on this, a list of 5 animals. And those animals you talked about, Egyptian fruit bats, onward transmission, deer mice, onward transmission, deer, but we all know about deer now, onward transmission, mink. What I thought were ferrets like you were actually mink. And they're not just, any kind of mink, they're American mink. And they're found, everywhere except China. None of these animals are in the Eastern Hemisphere, unnatural, or natural, I should say.
Yep.
Then the 5th one is the hamster. This one blew my mind. I thought for sure you would find a Syrian hamster in the Wuhan Institute of Virology. There's none. This is that little pet you had or your kids have as a, when they're a kid, and yes, and they live for a couple of years and you throw it away. It's just it's called a Syrian golden hamster, and they're not found in China because they have their own called a Chinese hamster, which doesn't really, it's not a transmission model for SARS-2. When I found out the 5th transmission model wasn't even in China, I'm like, wow. I mean, this is incredible. So I propose this to be our biological proof of a Wuhan area lab leak and a Rocky Mountain lab origin, because there's only one place on the planet before the pandemic where all 5 of those models that I listed are found, and that's inside Rocky Mountain lab. And pretty much, I'm not going to get into the list, but I do at the very end of the book, which ones were used for transmissible models on contagious vaccines, which is what they're developing in Rocky Mountain lab. They've been, talking and publishing about this in Rocky Mountain lab since like 2012, openly on transmissible vaccine research for all kinds of animals. And all these animals are basically only found there pre and post pandemic inside Rocky Mountain lab.
So I want to drill into the science of this a little bit. So the thing was sent to them, and they were working on enhancing maybe transmissibility of the virus. So that what does this mean? They pushed it through those 5 model species, and your contention would be that the thing that had been pushed through those 5 species was then the thing that got out in Wuhan.
Correct. And, it's not, a lot of people come at me about, was it the ancestral strain? Yes, it is. WA1 is the best known ancestral strain of SARS-CoV-2. WA1 stands for Washington Area 1. It's a beautiful story. It's chapter 27 in the book, America's Patient Zero. Maybe I should have called him Patient 1. I'm not actually sure how that works. But there's a man in his mid-30s, unnamed. The New York Times talked to him, but didn't name him, who traveled to Wuhan in middle of November of 2019. And he's obviously, I think, renewing a visa, and I'm guessing he works in the tech sector somewhere in Seattle. And he's there for about 6 to 8 weeks, and he's traveling back to the United States on January 15th of 2020. He never visited the wet market. He did go to Wuhan, though, obviously, during the pandemic, and unknowingly traveled back to the U.S. with the best-known ancestral strain of SARS-CoV-2. WA1, Washington Area 1, was the isolated from his lungs and sent off to the CDC and sent around to all these different, labs in the U.S. where we got to learn about what animal models transmit SARS-CoV-2 and which animal models do not. WA1 predates lineage A and B found in the wet market. I have eliminated the wet market from this equation, basically. It's essentially, out of the picture. It's only 3 nucleotides out of 30,000. But that's not a lot in the grand scheme of things, but it's still considered the ancestral strain of SARS-CoV-2 called Washington Area 1, WA1. And that ancestral strain transmits efficiently in those 5 animals we've talked about. So I consider that to be the biological proof of the Wuhan area lab leak and the US area lab origin.
Okay. I think that was very clear. I think people listening understand what your contention is and the logic that leads up to it. We've been on for quite a while, so maybe we should start wrapping up. I just want to say again to people who are listening, if you're interested in this, buy Jim's book, read it, think about it. I hope this conversation will stimulate further scientific discussion. I'm not an expert. I'm not taking a position on this, but I think, I'm happy to have the conversation. Jim seems to have a lot of interesting facts that I didn't have access to before I talked to him. Jim, if everything fell into place, if the HHS director coming in under Trump were your best friend, or maybe Trump or Elon was your best friend, what steps can be taken to really sort this thing out finally?
You can subpoena people and keep doing FOIAs, and they're going to keep doing these charades. It took us 5 years. Us is not me, it's other people. I did not file a FOIA. I did not write a research paper. I just stand on the shoulders of others. It took us 5 years to get to see these emails inside Rocky Mountain Lab. And just to put a footnote on this, there is a smoking gun email, in the book that essentially proves the Egyptian fruit bat is a transmission model. And I want to make this very clear because it exonerates Danielle Anderson of any responsibility for this thing. Real quick, Tony Shantz keeps the Egyptian fruit bat colony for Rocky Mountain Lab. He breeds them in captivity for Rocky Mountain Lab. DARPA pays him to breed them in captivity for Rocky Mountain Lab. DARPA pays Tony Shantz of Colorado State to send his bats to Hamilton, Montana once a year, $1,000 a year. So in a way, the DARPA, preempt team, which beat Ralph Baric and Peter Daszak, had a hand on this too. So, in that, exchange, I want to get to this because this is the smoking gun email, the smoke stick email. I put this in front of every bat virologist on the planet. Prove me wrong. I wish, and I would move on with my life gladly. But inside this email, essentially, is Tony Shantz out of curiosity. He's kind of the Limpa Wong of Colorado. He's the Batman of the U.S. Out of his own genuine curiosity, good guy. These are all interesting people with their own curiosity. I'm gonna stick every animal I got, and I'm gonna detect it. This is now February of 2020, and Vincent Munster of Rocky Mountain Labs says, "Don't do it. Don't do it. Myself and Ralph Baric have already tested that bat, and it didn't work." And Vincent Munster sends Tony Shantz an email with the link to this 2018 paper that we started this conversation off with. WIV1 does not infect Egyptian fruit bats. And so he's trying to discourage Tony Shantz from actually running the test in February 2020. Out of just joyful curiosity, Tony Shantz fires off the email back to him and says, we have some curious results. We have 21-day positive test results of my Egyptian fruit bat colony without symptoms. And you get this, Vincent Munster. Not only did my contact bats get infected, they're the ones that aren't, they're the virgin bats put in after the bat's been infected. A day after, you infect 3 bats, then you put in a contact bat in the same cage to see if you get transmission onto the next bat. That's very typical. Then he had a negative control sample housed in a cage above the infected bats. Above was the word he used in all capital letters. And he says, I can't believe it, but all my, negative control bats are infected by this thing too. This is really weird, isn't it, Vincent Munster? And he said he put a smoke stick up there to get the airflow from the HEPA filter down to the ground inside Colorado where he's at in Fort Collins. And it came around the other side and it infected this negative control bat cage above. This thing was so contagious. There was no containing it. This thing would have leaked from a BSL-5. They threw the kitchen sink at this thing, not to infect us humans. It's an accident. It's an animal vaccine to infect live bats. This thing, the R0 was cranked up. Everything about this thing, is designed to transmit through air, through aerosols. And that was shown in what I consider to be the smoking gun email between these two.
Okay.
So unknowingly gave it up.
Okay. Just to elaborate on that a little bit. So the actual virus you're talking about here is WA1. Okay, so this is an experiment done with WA1 February 2020 at Colorado State. Guy who's an expert with fruit bats. And it's found to be incredibly contagious among fruit bats. That email from Munster where he cited the failed result from 2018 with a different virus. What's the thought process there? He's telling his friend, don't try this in your bats, but is he saying it's not going to work because look at this paper, it didn't work in fruit. The other thing didn't work in fruit bats. What's going on there?
He's gaslighting them, kind of like Baric did Kristian Anderson. He's that's kind of that parallel I'm gonna get into. I mean, the email is in the book. It's public record now. It only emerged this past summer, meaning 2024. But it's incredible to have. I will admit this: inside the email, I personally knew what to look for. Now, I've been trudging through these FOIA documents, looking for something, because I woke up one day and I was like, you know what? The US CDC is sitting on unpublished data and I'm going to accuse them of it. And I'm like, well, why don't I just email the guy and ask him, hey, you keep an Egyptian fruit bat colony in Atlanta, Georgia, and me sitting in this chair in Houston, Texas, know that you're sitting on published data because I know SARS-CoV-2, WA1, infects Egyptian fruit bats. How do I know that? Because the Germans already showed that. So why don't you publish your results? He says, well, we don't want to repeat the results the Germans already tested, but it is a non-natural reservoir host. The non-natural reservoir host was the thing that stuck in my mind. I don't name the guy in the book. I'm not going to name him. I don't really care. But he gave me what I needed, self-confidence to look through these FOIA documents to find out what I thought. And as I'm reading through it, I read the paragraph where Tony Shantz describes the test, and I thought, that's Egyptian fruit bats. To their defense, I look at the subject line of the email, and it says Jamaican fruit bats. This is basically a New World lab animal, basically, that's used a lot. But back below it, you can see what's happening if you go all the way down to the February email. You can see that this is they call it the bat, Rosettas. That's essentially the Latin name for this Egyptian fruit bat. So it was hard to tell exactly which bat they're talking about. But I asked Tony Shantz directly, did you run this test on Egyptian fruit bats? I did it via Twitter. And then he deleted his Twitter account. So he's gone. I mean, he was accessible. Now he's not accessible. Wow. I've asked these people to provide me a defense, a denial. I asked Vincent Munster, did he run it? And if he did, why don't you publish the results? So I guess to answer your question that you started off with before I get off these tangents is, what can we do? We can educate ourselves on this thing. We can have an adult conversation about this thing. These people did not set out to blow up the world. They were trying to, infect bats. They wound up infecting us. If we just admit this and we go with them, who's we, as truth seekers, and we say, you, Rocky Mountain Lab, are sitting on unpublished data. You have not published the Egyptian fruit bat transmission data. Please publish that. Obviously, the test has been run, but it hasn't been published. Since then, Vincent Munster has tested Jamaican fruit bats, all kinds of other little weird things. He's run all kinds of tests on various bats, but he will not do the Egyptian fruit bat test. So he's sitting on that unpublished data. He just needs to give this to us, Let us humans, on this planet that endured this, see the results of the W1 transmission test. He has not done it on deer. Kansas did, but he has not. Kansas State University did, but he has not done. He did not do it on deer mice. Again, Tony Shantz did it on deer mice, but not Vincent Munster. And Tony Shantz has gotten mad at him in some emails. Don't you have a deer mice colony? Why aren't you running this test? Why do I have to do it? Why do I have to waste my funds? It's kind of funny to see these two go at it with each other. He did do the mink, but he did not do the mink, meaning the Neo-Bison bison. But I don't think he didn't do WA1. He's a bit sneaky when he does things. He may not use the ancestral strain. And then someone else in Rocky Mountain Lab did the Syrian hamster. So Rocky Mountain Lab, if we were to get together and say, look, let's have an open letter, open scientific discussion. You have lab animals inside your lab, pre-pandemic, that have not been tested with WA1 isolate, the best known ancestral strain of SARS-CoV-2. Please publish the data so that we can see the results.
Are you sure this would really prove the result? Because it's already known that it transmits in those species, right, from other labs. So
I just bring it up because it's a cover up, and what you can understand it being a physicist. I can understand because I've just thought about this way too much. But your average Joe on the street cannot comprehend the fact that, something cooked up in Montana could have been used in Wuhan and leaked and came out and blew up the world and they covered it up. But if you propose to, the people that don't really think about this too often, that look, they're sitting on unpublished data, they understand there is a cover up and there's something more to be said. And then maybe, there's a political leverage to go in there and open up the lab books. We the whole time everyone's been wanting to see the lab books of the WIV. We saw them when Xi Zhengli published RATG13. She opened up the lab book. She opened up the database. We saw her records, essentially. We saw Ralph Baric's records with the FOIA. I have a genome 20% different than SARS-2, or SARS-1, and that turned out to be SARS-2. Yeah. So that was their FOIA.
A little in the weeds question, because I'm not sure I understand the actual biology of this. So if I want to make it, suppose I had those 5 species, deer, mice, mink, hamsters, fruit bats, in my lab in Montana, and Baric has sent me this sample, and I'm trying to make it transmissible in those 5 species. What am I actually doing? Am I modifying the virus further to make it transmissible in those model species, or what do you actually do?
How does Munster aerosolize the agent? Is that what you're asking?
Exactly. Yes. Sorry.
So it's not public record. He was not allowed to publish how he did it. And he is basically the protege of Ron Fauci. So when you read about the engineered doomsday in 2012 from the New York Times, it was the Ron Fauci experiment of ferrets in Erasmus University in Rotterdam, the Netherlands. And the lead author on some of those papers was Vincent Munster. He is the protege of one Ron Fauci. And Ron Fauci was the face of that old disaster, along with Tony Fauci, who funded it. And there's a huge debate, along with give the Richard E. Bright's credit, the biodefense hawks, I would say that, the biosecurity hawks, did not allow Ron Fauci and Tony Fauci to publish the results. They did not publish the entire way it was done. So it was always an unpublished state secret how to aerosolize the agent. Best I can piece together, this is how it's done without rambling too much. I've listened to Fauci talk a lot. He calls it 3 phenotypes. You aim for the upper respiratory tract at a targeted temperature. Massive amounts of virus in the upper respiratory tract at a targeted temperature is the key there. As best I can tell, this bat, world, these bats actually ironically have about the same, body temperature in the upper respiratory tract, a similar ACE2 receptor as us in the upper respiratory tract. So they aim for these 3 phenotypes, a massive amount of virus in the upper respiratory tract at a targeted temperature. And you do it by taking the genome and you shove it down the nose of one, you force the sneeze, you get it into the air, and then you quickly bring in the contact animal to get the thing transmissible. And as you daisy chain it about 6 or 7 times over, by forcing the sneeze, you get this transmissible agent. And you also you can see Munster's. You start with a small animal, like the Syrian golden hamster, which the kids play with, and you have these tubes that are about a meter apart, or then they're 2 meters apart, then they're 3 meters apart. So he's trained the agent how to transmit further in a small animal, basically over time.
I get it now. So they're relying on the natural mutability, the natural variation of the virus, and then they're setting it up so that what they end up harvesting is the one that could actually traverse that.
He's trying to make the microns of the agent as small as possible. And that's one of those unique features of SARS-CoV-2. It's like 3 or 4 microns. I don't even know what it was, but it's so small it can float upwards of 60 feet.
So they're basically using the existing variation in the virus because it's very mutable, and then they're just basically selecting it for the ones that transmit through the air. So
there's a lot of these are Ralph Baric was very uncomfortable with transmission of coronavirus. To his defense, that's why he always used that mouse model.
Yeah,
he said he would feel icky. Immoral was a term. I don't remember what I used. I used it in the book.
Yeah,
he has a quote.
I understand. You want non-sneezing systems.
Yeah.
Because
That's exactly right.
Otherwise, somebody can use the sneezing transmission as a way to select for variants of the virus that then are aerosolized. So I understand. Okay. Jim, it's been great talking to you. I appreciate everything you've done. I feel like we've done a good job elaborating this so the audience who's willing to sit through 2 hours of this will come away with a deeper understanding. I did of origins of SARS-CoV-2. I just want to end by saying that I'm not taking any position on this. These are all real scientists with probably honorable motivations for what they were doing at the time. My base assumption is because there have been many zoonotic transmissions, that any new virus or pandemic, my base assumption would be it's probably zoonotic. Engineering is an extra step. So if you're a Bayesian, you might, reduce the probability a priori that it could be engineered. And so one should, have a high threshold for evidence that before one concludes that it was actually engineered. But of course, I think it's important to try to understand these things because this kind of research is clearly very dangerous and deserves more attention. So let me end there. Thank you again, Jim, for coming on the podcast.
Thanks for having me, Steve.